Happiness. I welcome you all for the Ardent MDS. The topic what we are going to see is about structure of DNA. So one of the very important topic, but some of the students during NEET preparation feel difficulty to answer those type of question comes from this topic. So this aim of this video is to make the lecture very easy and able to answer for any question if it comes from the structure of DNA. Right. So before we start with the DNA structure, you have to know the chemistry of nucleic acid and what is called nucleic acid. Nucleic acid is nothing but it is just a polymer of nucleotide. Polymer of nucleotide. And what is called nucleotide? Nucleotide is nothing but a nucleoside plus phosphate group joins together makes the nucleotide. And what is this nucleoside again? Nucleoside is nothing but nitrogen base plus sugar which makes the nucleoside. Again your question, what is nitrogen base? Example for the nitrogen base, purine or pyrimidine is an example for the nitrogen base. Ribose and deoxyribose is an example for sugar. Both ribose and deoxyribose are pentose sugar. In case of RNA, ribose sugar is present. In case of DNA, deoxyribose sugar is present. Right. Example for purine, adenine and guanine, which are example for purine, cytosine and thymine in case of DNA. In case of RNA, uracil is present. Right. So again, I just repeat one more time. Nucleoside plus phosphate group, phosphoric acid, joins together makes the nucleotide. All nucleotide joins together makes the nucleic acid. And what is called nucleoside? Nitrogen base plus sugar. Example for nitrogen base is purine and pyrimidine. Example for purine is adenine and guanine. Example for pyrimidine is cytosine and thymine. Now all together, this nucleotide which makes the DNA, this is how it looks like. The structure of DNA, a model of DNA which is given by a person called Watson and Crick. In the year of 1953. Specifically, this is a model called as type B model, as called as right-handed helix. Right. So now seeing the structure of the DNA. DNA is a double helix structure, you all know very well. So that is a question which was asked in one of the exam. Collagen is a structure of double helix, triple helix or something else. Collagen is a structure of triple helix. DNA is a structure of double helix. Why it is helix? Why it is coiled with each other? It can be straight, right? That is the reason for this. Because the nucleus is only few micrometer, but DNA is length of 2 meter. Okay, so we cannot pack the 2 meter into a small nucleus. The DNA is present inside the nucleus, right? So to accommodate the DNA into the nucleus, it became coiled so that it can able to present inside the nucleus. There are some DNA which present in mitochondria, which is responsible for the mitochondrial inheritance. So this is how it look like. So as like we studied in the previous slide, so this is called nitrogen base. Nitrogen base which joins together with the sugar to form nucleoside. The nucleoside with the phosphoric acid to make nucleotide. Clear? So coming to this nucleoside, always the nitrogen base, the purine joins with the pyrimidine, adenine joins with thymine, guanine joins with cytosine. If you see this between the adenine and thymine, there are 2 hydrogen bonds. In between the guanine and cytosine, there are 3 hydrogen bonds. So this is one of the questions they may ask you. How many hydrogen bonds present between the adenine and thymine? How many hydrogen bonds present between the guanine and cytosine? And in between this sugar molecule, a base molecule, van der Waals bond is present. Here hydrogen bond is present. And the bond between this nucleoside and sugar, this is a deoxyribose sugar. So deoxyribose sugar is a pentose. If you see the structure, you can able to understand it is a pentose sugar. So the bond between this nucleoside mean nitrogen base and this pentose sugar, which is a glycosidic bond. So there are three types of bond. Here a glycosidic bond, here a van der Waals bond, here a hydrogen bond. And in between this nucleotide, every nucleotide, okay, the nucleotide which joins together by phosphodiester bond. Phosphodiester bond. So there are four types of bond which you have to know. In between the purine and pyrimidine, you have a hydrogen bond. Number one. Number two. In between the base, you have a van der Waals force. In between the nitrogen base and a sugar, glycosidic bond. In between the nucleotide, you have a phosphodiester bond. Clear? And what else you have to know? Apart from this, what is the diameter of the DNA? Diameter of the DNA is around 20 angstrom. And suppose this is because the double helix structure. So if you see 4 plus 4, 8, 8 plus 2, 10. So this 10 base pair which all together makes a one complete turn of the DNA. One complete turn which is called as pitch. So the distance or length of the pitch is around 34 angstrom. When it is coiled, it makes two grooves, that is one major groove and a minor groove. The length of the major groove is 22 angstrom and the length of the minor groove is 12 angstrom. If they want to ask you some numerical type of MCQ, they may given this type of question. And in between the two bases, the distance is 3.4 angstrom. Clear? Diameter is 20 angstrom. And how many base pairs is equal to one complete term? 10. 4 plus 4, 8. 8 plus 2, 10. So 10 base pairs equal to one complete 10. One complete 10 is called as pitch. The length of the pitch is around 34 angstrom. And when it is coiled, it forms two grooves. One is a major groove. The other one which is called as minor groove. Length of the major groove is 22 angstrom and length of the minor groove is 12 angstrom. Clear? Right. Now you know what is called nucleoside, what is called nucleotide, and what is called nucleic acid. And there is now one special terminology called as nucleosome. What is this nucleosome? Right? So now you know what is called nucleotide, right? This nucleotide which joins with a special protein called as histone. So this together makes this nucleosome. And histone have several type of protein like H1, H2A, H2B, H3 and H4 are the subtypes of histone. And two molecules of H2A, two molecules of H2B, two molecules of H3 and two molecules of H4. So all together 8. So 8 molecules of histone except H1. 8 molecules of histone plus a nucleotide which coil with each other, makes the nucleosome. Clear? So histone does not participate here. So exactly this is look like this. So if you see this, there are 8 molecules of histone. So because it is 8 molecule, we can say octamer, histone octamer. So nucleotide is coiled with this histone which makes the nucleosome. In this nucleosome, the support for the nucleosome is given by the extra histone called as H1. So H1 histone which makes the support for the nucleosome, that is called as link DNA. So now this polymer of nucleosome, all nucleosome together which makes chromatin, So DNA which surrounding this histone molecule which makes these are all chromatin. all chromatin again condensed together, which makes this chromosome. Clear? Now, there are two types of chromatin. One is euchromatin, the second type which is called heterochromatin. Euchromatin is, if you see this, it is an uncondensed form of chromatin which is called as euchromatin, and a condensed form of chromatin which is called heterochromatin. Okay, so euchromatin is a starting stage. Heterochromatin is a end stage before chromosome formation. Now there is a confusion between three terminology for the student. What is called chromatin, what is called chromosome, what is called chromatin. Right. So what is chromatin? Chromatin is uncondensed form. Uncondensed form of chromosome. Okay. Chromatin all condensed together makes the chromosome. So now if somebody asks you what is called chromosome, now you can say condensed form of chromatin which is called chromosome. And condensed form of chromosome is called as chromatin. Condensed form of chromatin is called as chromosome. Easy, right? I know what is called chromatin. Yes. Chromatin is half of chromatin is called as chromatin. So now you know what is chromatin, what is chromosome, what is chromatin, right? So now we know what is the difference between euchromatin and heterochromatin. And the last step is about what is called denaturation of protein. By using the heat or some chemical like alkali, okay, you mix the separation of double helix. Separation of double helix into single strand by a process called as denaturation. So at the particular point where the helix separated, it is called as Tm, that is called melting temperature. Melting temperature of chromosome or DNA is around 85 degrees Celsius. Right. When the strand is separated, it is called denatured. So it has a special property. The special property is it absorbs the UV light. So that is number one. Hyperchromacity. Hyperchromic. Number two, viscosity. What about the viscosity? When it's separated, it's more flexible, so viscosity will be decreased. So these are the things which you have to know for the structure of DNA. Alright? So what are the things which you have to know for this take home message in the structure of DNA? I just tell you one by one. Right? Who given the structure of DNA? Watson and Crick in the year of 1953. Number two. What is nucleic acid? DNA is deoxyribonucleic acid. Okay, what is nucleic acid? Polymer of nucleotide. What is nucleotide? Nucleoside plus a sugar which makes the phosphoric acid which makes the nucleotide. And example for the nitrogen base, purine and pyrimidine are the example for the nitrogen base. Example for the purine, adenine and guanine. Example for pyrimidine, thymine and cytosine. Right? And what is called major group, what is called minor group. So during the coiling of the DNA, there are two groups will be produced, major group and minor group. Major group is a lengthy one and minor group is a smaller one. What is the diameter of the DNA? 20 angstrom. So this nucleotide which coils with a special protein called as histone makes the nucleosome. The polymer of nucleosome which is called as chromatin, and the chromatin is condensed together makes the chromatin. Okay. So chromatin is uncondensed form of chromosome. Chromosome is condensed form of chromatin. So again, how many histone molecules will be used for making this chromatin? 8 molecules. With that, one strand of H1 histone will be occupied, which gives the support to the nucleosome. And there are two types of chromatin, euchromatin and heterochromatin. Euchromatin is a completely uncondensed form and heterochromatin is a condensed form. Right? So what is called denaturation of protein? Separates the strand apart. That is called as denaturation of protein. The particular temperature where it occurs is called as TM, that is melting temperature. So these are the things which you have to know in your mind before answering this multiple choice question coming from the structure of DNA. My dear aspirants, so these are the few questions which was designed by us for the structure of DNA. Hopefully this will be useful for you. So the first question is about Watson and Crick model is for which type of DNA. So you all know very well the DNA is a double helix structure which was given by Watson and Crick in the year of 1953. And there are 6 types of DNA: A, B, C, D, E and Z. Among this, the original model which was given by Watson and Crick is B type of model. So answer for this is first option. So we can go for A. BDNA. And what is about this BDNA? It is a right-handed helix or left-handed helix? The BDNA is a right-handed helix. Right. Question number 2. First carbon of pentose sugar of nucleic acid joins with. Right. So to answer this question, you have to know the structure of DNA. So if you see this, so this is the ribose sugar. So ribose sugar is a pentose sugar. That means it has a 5 carbon. So among this, the first carbon. So the first carbon which joins with which nitrogen of the pyrimidine or purine. So this is the question which was asked. So the answer for this is second option. So the ribose sugar, C1 of the ribose sugar which joins with N1 of the pyrimidine or N9 of the purine. So these are the things which you have to know. And what is this bond called as? This bond is glycosidic bond. Right. Question number 3. The 2 strands of DNA are held together by. Right. So these are the 2 strands of DNA. So in between the 2 strands of DNA you have something called as hydrogen bonds. You have something called as hydrogen bonds. And how many hydrogen bonds are present? Again, this may be asked in your MCQ. In between adenine and thymine. So adenine belongs to the purine, thymine belongs to the pyrimidine. So in between adenine and thymine, there are 2 hydrogen bonds. And in between guanine and cytosine, there are 3 hydrogen bonds. Right. So what are the other bonds which present? As I told you, the bond between the ribose sugar and the purine is glycosidic bond, and 2 bases. These 2 bases which joins together by van der Waals force. Number 3. Number 4. 2 nucleotides joins together by phosphodiester bond. So these are the various type of bonds present here in the DNA structure. Question number 4. At physiological pH, DNA is. What is the answer? DNA is negatively charged, right? Acidic, right? So why we can go for an acidic? So because the DNA is a deoxyribose sugar attached with nucleic acid with a phosphoric acid. So this phosphoric acid, so we can go for acidic. So phosphoric acid is a negatively charged, so we can go for a negatively charged molecule, right? So what about this amphipathic? So amphipathic is, if a structure has both polar and non-polar group, it is called as amphipathic. Right. So coming to here, so inside of the DNA structure, so it is a non-polar, and outside the structure it is polar. So it has a polar and non-polar group here. So we can tell it as amphipathic. So go for the all of the above. Right. Question number 5. Very important thing. What is Chargaff's law? Chargaff's rule is quantity of purine is equal to the quantity of pyrimidine. So you know very well, adenine and guanine, which are purine, thymine and cytosine which are pyrimidine. So both are equal. Okay, quantity of purine is equal to quantity of pyrimidine. Mean adenine is equal to thymine, guanine is equal to cytosine. Right, so we can go for this option. Right. And question number 6. On complete hydrolysis of DNA, we'll get all the following except. On complete hydrolysis, the whole DNA just break and produce urine base, phosphoric acid, pentose sugar, and it also produce this nitrogen base called adenine, not adenosine. So this one is the answer for question number 6. And so one more way to answer for this question is DNA. They are asking about DNA. So on complete hydrolysis, it forms a deoxyadenine. So this given as adenosine. So that way also I can eliminate this option. So go for the first option. So keep in mind this adenine is from adenine, guanine, cytosine, thymine. So not adenosine. Right. Question number 7. Which of the following is not a pyrimidine? Right. So just now we studied about adenine and guanine which are purine, thymine, cytosine. In case of RNA it is uracil also there. So thymine, cytosine, uracil are belongs to the pyrimidine. So thymine, uracil, cytosine are pyrimidine. So adenine is a purine base. So for the 7th question you can go for the 3rd option. Right, question number 8. What is the true about DNA structure? Yes, adenine and guanine are purine, right? Uracil and cytosine are pyrimidine, right? Watson and Crick, child. True about DNA structure. Purines are adenine and guanine, right? And pyrimidines are uracil and cytosine. So it is thymine. In case of DNA, it is thymine, not uracil. Uracil will come for RNA. So cytosine and thymine present here, not uracil. So first option is the wrong option. Question number 2nd option. Watson and Crick discovered the DNA structure in 1953, not 1973. So we can able to eliminate this structure. Mainly consists of left-handed helix. The original structure of DNA which was given by Watson and Crick in the year of 1953, and it belongs to the right-handed helix, not left-handed helix. So you can eliminate this. Deoxyribose backbone with the bases present on the inside is right. So bases are present. Like if this is the structure of DNA, the bases are present inside. Okay, and the backbone is about this ribose sugar and the phosphoric acid present in the backbone of DNA. So this is right. So you can go for the 4th option. Right. So it's very easy to answer once you are able to understand the concept and some of the few facts which given in my video about structure of DNA. Then you can able to answer any type of question from here. Okay, hope this video is useful for your competitive exam, and we are very happy to help you. Thanks for watching.